Alrighty, second video in the kind of realm of targeted transgenesis. And we'll kind of start this one off calling back to the knockout mouse that I talked about in the last lecture. So this was the ability to go in and specifically disrupt the expression of a gene you're interested in studying. Okay, so this calls back to the idea of reverse genetics, this being the overall strategy of disrupting the function of a gene and then seeing what kind of a phenotype that produces, and then using that phenotype you can then infer or try and figure out what it is that gene does. Like I said, a very fundamental strategy in really biology as a whole. But as I want to say a lot, if you ever see me in class again, you'll probably hear this 1,000 more times, but there's no such thing as a perfect experiment. There's no such thing as a perfect technique, a perfect strategy, perfect approach. I think there's some value in sort of embracing that in kind of the broader world as you kind of make your way through it. There's never going to be a perfect answer. There's never going to be a perfect solution. I think there's no better way to illustrate that than really what we're kind of going through right now. Thinking in terms specifically of how Luther's going to deal with the fall. There is no perfect solution for us. We're going to kind of do our best to sort of do what we think is right, maybe what we think is going to be the best for us, but again, fully acknowledging that there's no such thing as a perfect solution to this one. All right, so back to genetics. This knockout mouse, it, like anything else, has its limitations, right? And I teased this in the last video, and talking about sort of one of the major challenges that you might run into if you make a knockout mouse happens to be whether or not sort of the overall, it happens to deal with the overall importance of the gene you're trying to study, okay? So one consequence is that sometimes eliminating expression of a gene let's think back to one of the earlier single gene exceptions to Mendelian inheritance. Okay, this is this idea that certain genotype combinations could actually be lethal. So one of the more common ones is recessive lethal. Okay, so if a recessive allele produces a non-functional protein product, you can kind of think about that as a knockout allele, right? If it's non-functional, then it's really kind of a knockout. So sometimes, depending on how essential that gene product is, say that's a very essential protein like actin is something that's in your cytoskeleton, that's a pretty essential protein. And so two recessive mutations or two recessive alleles could be lethal in that case. So that's recessive lethality. And this is somewhat similar to this. So instead of kind of studying mutations that have arisen over time, we're kind of purposely engineering a mutation in the gene to reduce its function. And so it's entirely possible that by eliminating expression of the gene, you're going to kill the mouse, right? And so if you're trying to study a mouse, it doesn't help if that mouse never grows up and develops. Okay, so one possible workaround for this is that sometimes you can maintain your knockout mouse. Okay, so KO is an abbreviation for knockout. So you can maintain your knockout mouse as a heterozygote. Okay? So instead of, so basically having one wild type allele and one knockout allele. Okay? And so this could be interesting, especially in the case if this knockout allele ended up producing a situation in which these two alleles were incompletely dominant. So you've reduced the sort of amount of protein produced in this mouse from this gene by half. Sometimes that can produce a phenotype, especially if this gene is really essential. Sometimes this lethality can arise at different developmental points. So sometimes you can take two heterozygotes, mate them together, and you can study a fraction of those offspring. So if we have a wild type, if we take two heterozygotes and mate them together, you'd expect that a quarter of those progeny would have two knockout alleles. You can do the Punnett square if you want to. but maybe that recessive lethal or that knockout lethality doesn't arise until those mice are maybe just after they're born, or maybe they kind of live for a week, and you can kind of study them up to that point. So that's one possible workaround for this whole thing. You can kind of maintain your strain as a heterozygote strain and avoid some of this lethality associated with completely eliminating gene expression, right? So that's one workaround, okay? But the other sort of workaround that I'm going to talk about now is what's called a conditional, let's see, we'll call this first workaround. We'll call this one the second workaround. Okay, and this involves creating what's called a conditional knockout mouse. Okay, conditional being the kind of important term here. Okay, so in this case, a conditional knockout mouse allows you to regulate when and where gene expression is eliminated. Okay. So this is, I mean, at least conceptually, this is a pretty powerful idea, right? So this kind of gives you a lot of flexibility in terms of kind of designing an experiment to analyze the function of a gene, all stemming off of that basic idea of studying the function of a gene by eliminating expression. So this kind of some kind of a strategy that allow you to maybe eliminate expression of your gene in a tissue of interest. Let's say your gene is you're particularly interested in the role that your gene plays in muscle function. Okay. If you're going to study this in a mouse model, you might be able to conditionally delete this gene just from muscle cells. And that may work around this idea that if you completely eliminate it in all the tissues, that's lethal. And you could sort of use similar strategies for maybe deleting expression of this gene in the skin or the eyes or the brain or something like that. And you also then kind of this idea of being able to control when a gene is eliminated will give you some flexibility in terms of maybe I want to study this gene in an adult organism rather than a developing organism. So what if I can kind of tweak my system such that the gene won't be eliminated until the organism, let's say the mouse I'm using, grows up to about 2 or 3 weeks old. That was kind of if you're a 3-week-old mouse, you're effectively an adult, right? So there's a lot of experimental and flexibility that you could kind of generate from this kind of a system. And so this system does exist, and it is called a Cre-Lox system. Okay, so this is I'll redraw that one. So Cre-Lox system. All right. So what the heck is a Cre-Lox system? That's what we'll kind of talk about now. Right? Cre-Lox system. So really the key to this is this protein called Cre. Okay, so Cre is an enzyme, and it's actually a recombinase. Okay, so going off of what we know about how enzymes are named, the ACE means it's an enzyme, and the thing right before ACE is what it does. So this is an enzyme that catalyzes recombination. Okay, it is derived from, I think it's the P1 bacteriophage. So it's another nice example of going out into the world, studying the basic biology of a lot of different cells, and sort of co-opting that technology for the use in a research setting. Derived from P1 bacteriophage. Now, importantly, it's what's known as a site-specific recombinase, which just means that it's going to recombine DNA at very specific sites. Okay, and that brings us to the other part of this Cre-Lox system. So the site are what are known as LOX P sites. Okay, so It's Cre-Lox. Cre is the recombinase. Lox is this sort of sequence in the DNA. So by site, I mean this is an actual sequence in the DNA. Okay, so the way this works is if you've got a stretch of DNA right here, and within this you've got what are known as Lox P sites. They're often diagrammed as big triangles. Let's say you've got a section of DNA in here you're trying to get rid of. These Lox P sites occur in tandem, so there's always two of them. Okay. So these are the Lox P sites. Here and here. Okay. And so the way this Cre-Lox system works is that if you then take this system and you introduce Cre, which is a protein, it's a recombinase, it's a site-specific recombinase, it's actually going to recombine out the DNA in between the Lox P sites. So you end up with something that looks like this. So a single Lox P site is left in the genome. And then this intervening DNA is lopped out as a loop. Okay? So there's going to be a single Lox P site in that loop. And that stuff that was in the middle of it is eliminated. Okay, so this is Cre-Lox mediated recombination, or Cre mediated recombination. Okay. So you can start to imagine where if you get clever in the way you make your transgenic mouse strains, if you can start to engineer mice with these Lox P sites in them, that gives you some real flexibility in terms of what you can do in terms of studying gene expression, right? So just to clarify, this intervening loop oftentimes just gets degraded. So really all that's left in this genome then is this single Lox P site where this entire chunk of DNA used to be. Okay. So how does this look like as an overall strategy? Okay, so it's a little bit more complicated than just making a simple knockout mouse. But it's usually worth the payoff to put in the extra effort kind of at the forefront of the experiment to generate the final reagent, which is this mouse, okay? So this requires making 2 transgenic mouse strains. Okay? So mouse number 1 is known as the Lox mouse. Okay, so this is going to be a mouse into which you engineer these Lox P sites. Okay, so let's go back to our example part of the genome that has 4 exons. 1, 2, 3, and 4. And we'll go with our same overall strategy or goal, which is to eliminate expression of this gene. Okay. Again, there are some limitations to this, specifically relating to the size of genes in mammals. You can't put these Lox P sites too far away from each other, or else they're never going to find each other during this Cre-mediated recombination. So you have to actually have them somewhat close together for this to work. And so you do end up sort of targeting portions of this gene to affect its expression. So in the example I showed before where we inserted that neomycin cassette in place of one of the exons, we targeted exon 2. So for this one, we can imagine using a similar strategy by targeting exon 2. But in this case, the way we're going to do one first step of doing homologous recombination, we're going to engineer a transgene that has homology arms in these regions here, and then we insert into our transgene our Lox P sites flanking just the regular old normal exon 2. Okay? So this is the genome, and this is our transgene. Okay, so when this undergoes homologous recombination, we end up producing one LOX-P site, exon 2, LOX-P site, exon 3, exon 4, so on and so forth, right? Okay? So we've engineered this mouse, but the idea here is that the expression of this gene is normal until you actually introduce Cre into the system. Okay, so this is sort of, this LOX mouse now is dependent on Cre to actually then undergo the next step. Okay? Yeah, so this gene hopefully is functional. Now that's a big if. And so you actually have to go in there, and after you make this mouse, really make sure that by putting these LOX-P sites around this exon, you haven't disrupted the expression of this gene. Okay, remember back to this kind of running theme about how there are a lot of different ways to affect gene expression, not just sort of the coding region, and certainly there are a lot of different non-coding regions that can affect expression of the gene. We didn't go a whole lot into the details of what kind of happens in these intronic regions, but you can imagine if you affect splicing of this gene by somehow putting these LOX-P sites in the wrong place, you can affect the way that this gene gets expressed. There can be a lot of other ways that intronic sequences end up regulating expression of this gene or other genes. Sometimes enhancers for other genes can be located in introns of this gene, so you got to be really careful about that. Okay, so this is now what's informally known as a floxed mouse. F-L-O-X-E-D, floxed. Okay, so that's mouse number 1. Okay, the second mouse you need to make, you can probably guess, is your Cre mouse. Okay. Now this mouse is actually where things get interesting, and so this is going to be the source of the conditional specificity. Okay, so this mouse is going to be how you make things conditional. Okay, and so let's think back to this key for this whole thing, and that's that Cre-mediated recombination only occurs where and when Cre is expressed. Okay? And we have some control over that. Okay? So how do we control where Cre is expressed? Okay? We've seen some similar ideas to this. We can do this by honing in on promoters. Promoters and the regions kind of around them, including those cis regulatory sequences that we've been talking so much about in the McGregor et al. Paper, those regulate gene expression. They regulate the spatiotemporal expression profile. So what we're after then is trying to create a spatiotemporal expression pattern of Cre that can then recombine our floxed mouse wherever Cre is expressed. Okay, that's going to be how we generate the specificity here. Okay? And so this does require some work. You actually have to go in and characterize many promoters. Now, fortunately, there's a lot of people that do work on that. If they want to kind of study, say, a particular gene, they sort of take that promoter and they can make transcriptional reporter constructs. Again, similar to what they're doing in the McGregor et al. Paper. And you can take that knowledge from what they do and then say, okay, that looks like that gene is expressed in this tissue type at this developmental time point. Let me take that promoter, let me put that in front of Cre, and now I can drive Cre with that particular expression pattern. So that's kind of a good example of how seemingly disparate research areas can actually kind of talk to each other a little bit and how you can build off of someone's work. You know, they're studying a gene that has something to do with some biological process that you have no interest in, but what you're interested in is the promoter itself and the expression pattern of that particular gene. Okay? So let's just a quick diagram of what this looks like, and it's just to make it really simple. Let's say we're working with, let me, actually, I'm going to clear this out here. I suppose that's okay because you can just hit rewind on YouTube. Okay, so I'm going to use for an example of this, of a Cre mouse. I'm going to use what's known as an EMX1 Cre mouse. I won't go into a lot of details, but I'm going to use this because this is actually a mouse strain that I did some work with in my PhD. Okay, so in my PhD, we were interested in a gene called brain-derived neurotrophic factor. BDNF for short. It's a really important signaling molecule in the brain. It's really important for brain development, also important for things like for learning and memory. So in order to better understand the role of BDNF, we wanted to knock that gene out and then see what happens to brain development and brain function. Making a full knockout mouse of BDNF is lethal. So these mice actually, they die shortly after birth, okay, which necessitates making some kind of a conditional knockout mouse. And so we're particularly interested in studying the role of BDNF in the brain and specifically within the cortex and the hippocampus. So we wanted to study BDNF in a region called the forebrain. Okay? And so before I actually got in the lab, they did all the groundwork of trying to establish this system. And so the former graduate student in this lab went through and tried to characterize a bunch of genes to find genes that are expressed specifically within the cortex and the hippocampus, the forebrain area, starting from an early embryonic time point on. Okay, so this then led her to looking at this gene, so the promoter gene is a gene called EMX1. So what they did is they took the promoter of EMX1 just the promoter, not the coding region, just the promoter of it, and then put this upstream for regulating the expression of, we'll do it in green, how about that? Put this upstream then of Cre. Okay? So in this mouse, wherever EMX1 is expressed, Cre will be expressed as well. Okay? And so there are a variety of methods to make this Cre mouse, kind of ranging in specificity. One way to do this is to take something like this and just insert it randomly into the genome using something more akin to what you'd see in the transgenic plants, sort of some random integration of this transgene throughout that can have its limitations. This particular mouse, this EMX1 Cre mouse that we made in this lab was much more complicated, much more clever. It actually inserted this Cre open reading frame into the endogenous or normal EMX1 locus so that you could actually express both EMX1 protein and Cre using the iris that we talked about a while ago, this internal ribosome entry site. So it's a really clever strategy. If you're curious about it, shoot me an email and I can send you a paper where they describe making this EMX1 Cre mouse. It's a really nice example of really clever and well thought out genetic engineering. But the end point was that whenever EMX1 is turned on, you get Cre expression. Okay, so you end up making this Cre mRNA, and then you translate Cre into protein, and so then you've got Cre, which can then cause Cre-Lox or Cre-mediated recombination at these LOX-P sites. Okay. So once you have your two mice, so mouse one here, mouse one, I kind of erased the first part here, that leads you to the final step. got your Cre-Mouse. Actually, I should make this one green to be consistent. And you also have your Lox mouse. So the final step is to mate these together, to breed them. Okay? So then you'll get out of this progeny that are floxed, but then also express Cre in whatever sort of whatever expression pattern that you wanted. Okay? So in the case of EMX1 Cre, this was embryonic forebrain expression. And that's just one example of many. Okay? In fact, later on, they ended up using a different Cre driver, which still expressed Cre in the forebrain, but didn't turn on until much later. So this idea of what if we don't eliminate gene expression of BDNF until the mouse becomes an adult? And so for that one, they used a whole different promoter. They used one from a gene called CAM kinase 2 or CAMK2. So again, I'll kind of hint on this flexibility later. But this is that last step. So you take your Cre mouse and you you breed with your Lox mouse, and the progeny from this cross should have both Cre expression and Lox sites. So you should get then Cre-mediated recombination at your gene. So again, what does that look like? Well, this was kind of our Lox P mouse. Exons 1, 2, 3, 4. Here are our Lox sites here. Okay. You breed them together, we get Cre expression. It's then going to recombine the DNA in between these two Lox sites. And that results then in a gene that looks like this, a single Lox site. And the elimination of exon 2. Okay. Okay. Now what this usually does is this disrupts, we'll put an asterisk here, continue that up here. This disrupts the reading frame. And usually it does this in a way that actually introduces a premature stop codon. Okay, so normally if the reading frame is maintained through exons 1, 2, 3, and 4, by taking exon 2 out, usually you disrupt that reading frame, so those sort of ordered codons of 3 nucleotides. If you disrupt that at all, then you change the reading frame, and typically what that does is it results in the introduction of a premature stop codon, and the production of a non-functional truncated protein. Okay. Non-functional, i.e. truncated protein product based off of the introduction of some sort of premature stop codon. So the stop codon doesn't normally exist in exon 3 because the reading frame starts over here. But if you change that by omitting exon 2, you oftentimes introduce this premature stop codon. And then you end up getting no protein product. But that's only happening when and where Cre is expressed. Okay, so this is Cre-Lox mediated recombination. This is a way to make a conditional knockout mouse. Okay, so I don't even erase here, I think. I'll just kind of put it over here on the side. So one of the kind of big advantages to Cre-Lox systems is that once you have your Lox mouse, you can breed your Lox mouse with different Cre strains. Okay, so the example I have here was breeding it with an EMX1 Cre strain. But I also talked about how they went on after doing this EMX1 stuff to then breed this with a CAM-K2 Cre strain. Okay, similar spatial temporal expression, except that this CAM-K2 promoter didn't start expressing Cre until later on in adulthood. Okay, so they only had to make this Lox mouse once, and then you can go through and made it to different Cre driver strains. I call them Cre driver strains because it's the promoter here that's driving expression of the Cre recombinase. So that's a major advantage to this whole system, and so you can test different developmental time points when this gene turns on, different tissue expression patterns, all that kind of stuff. And really all this then really depends on the promoter here that's driving your Cre expression. Okay, kind of highlighting this sort of big key issue we talked about in gene expression. Promoters regulate when and where a gene is expressed. All right, I think that's all I've got for this one. Look for an email later today. Today is Tuesday, kind of updating where we're at in the class in terms of what's going to happen. I'm going to try and post a couple lectures tomorrow talking about kind of the next level in the specificity of genetic engineering, and that's going to be talking about CRISPR-Cas. I'll probably post that in a 2-video set. The first video will focus on the normal role of CRISPR-Cas and how it was discovered, and then the next video will talk about how CRISPR-Cas is used to do things similar to what this can do, but in kind of a more precise manner. All right, hope everyone's doing all right, and I'll see you tomorrow.